an image like no other, inspiring decades of research and debate. The Shroud of Turin is a remarkable piece of history, and it continues to challenge science with its unresolved mystery. Deep within the fibers of the Turin Shroud, scientists discovered something unexpected: human DNA. Advanced genetic analysis revealed mitochondrial DNA from several geographic regions, suggesting the cloth was handled by many people across different centuries. Yet some genetic fragments appeared unusually ancient, raising questions that science still cannot fully answer. For millions of believers, the shroud is thought to be the burial cloth of Jesus Christ. For scientists, however, it remains one of the most fascinating forensic mysteries ever investigated. What exactly did the DNA reveal, and why did it leave researchers so surprised? Subscribe for more mysteries uncovered through science. Skeptics see the story very differently. To them, the shroud may be the most convincing medieval forgery ever produced. Some even suggest Leonardo da Vinci could have created it. Others argue that it was the work of an unknown artist, carefully designed to attract pilgrims who longed for signs of miracles. For more than 600 years, the debate has continued. Faith stands on one side, reason on the other, and neither has ever truly surrendered its position. But in the 21st century, speculation alone was no longer enough. Science entered the discussion with powerful tools and precise methods. Genetics, high-energy physics, spectroscopy, and forensic science made it possible to investigate the cloth in ways that were once unimaginable. Researchers stopped treating the shroud only as a sacred object. Instead, they began examining it as if it were evidence from an unsolved case. In many ways, it became like a biological archive, quietly preserving traces of events that may have happened nearly 2,000 years ago. Scientists extracted and sequenced DNA trapped within the fibers. They scanned each thread using X-ray imaging. They analyzed the bloodstains at the level of molecules, atoms, and elemental structures. Initially, researchers expected simple explanations. Perhaps the cloth would reveal the DNA of a single individual, or maybe traces of pigment from a medieval artist who had painted the image. But the results turned out to be far more complex. Genetic specialists at the University of Padua, along with physicists working in the controlled laboratories of ENEA, discovered findings that surprised even the most experienced researchers. The data didn't simply challenge existing theories, it shattered them. Both skeptical explanations and traditional religious assumptions began to collapse under the weight of the evidence. The shroud suddenly appeared to be far more than just fabric. It looked like a map that traced a journey beginning 2,000 years ago, a record of suffering described with such detail that it has never been successfully reproduced, and perhaps even a sign of an intense burst of energy, one that modern physics still struggles to explain. This discovery marked the beginning of one of the most detailed forensic investigations ever attempted. Scientists began following clues invisible to the naked eye: microscopic pollen grains, chemical residues, and biological traces embedded in the cloth. Step by step, they tried to determine whose blood had soaked into those fibers, and why the biochemical evidence seemed to reflect extreme physical trauma. Researchers also studied the strange image imprinted on the linen. The image behaves in ways that ordinary artwork does not. It functions simultaneously like a photographic negative, a three-dimensional projection, and something resembling an X-ray. Another surprising discovery followed. DNA analysis suggested the artifact could not easily have been created in Europe. Instead, the genetic evidence pointed toward a more complex historical journey that may connect to events that shaped human history itself. To understand why these modern discoveries shocked the scientific world, we must return to the very beginning of serious shroud research, what is known today as modern syndenology. That story begins with a moment when humanity first clearly saw the face hidden within the shadows of the linen. The date was May 28, 1898, in the city of Turin. A lawyer, city council member, and passionate amateur photographer named Secondo Pia was given rare permission by King Umberto I to photograph the relic during a public exhibition celebrating the wedding of the royal prince. At that time, photography was anything but simple. It required large equipment, careful preparation, and long exposure times. Pia carried a camera, almost the size of a suitcase, and climbed tall scaffolding inside the cathedral, preparing to capture an image that would soon change the entire investigation forever. Inside the cathedral, the lighting was dim and difficult for photography. To capture the image, Secondo Pia relied on powerful magnesium flashes and bright lamps to illuminate the relic. He exposed two large glass photographic plates, each measuring about 50 by 60 centimeters. Later that night, back in his small, dark room, Pia began developing the photographs. The room was lit only by the faint red glow of a lantern as he carefully lowered one of the glass plates into the chemical solution. Then something unexpected happened. As the image slowly appeared on the glass, Pia reportedly froze in shock. The negative nearly slipped from his hands. What he saw felt almost unreal. On a photographic negative, light areas usually turn dark and dark areas turn light. But on the glass plate before him, the faint and blurry figure visible on the cloth suddenly transformed into something entirely different. Instead of a vague stain, the negative revealed a sharp and highly detailed positive image of a human face. It showed closed eyes, a broken nose, a mustache and split beard, and bruising on one side of the face. For the first time, the figure hidden in the linen was no longer a shadowy outline. It looked like the face of a real person. The expression appeared calm and composed, almost regal, yet the injuries suggested that the man had endured severe physical suffering. The discovery stunned Pia, but more importantly, it changed the way people understood the Shroud of Turin. Normally, if a drawing or painting is photographed and turned into a negative, the result looks distorted and unnatural. Light and dark areas reverse in a way that flattens the image. But the shroud behaved differently. When photographed as a negative, the figure on the cloth looked like a realistic black and white photograph of a human body. That single observation raised a huge question. The image visible to the naked eye already behaves like a photographic negative. So how could such an image have been created centuries before photography even existed? If the shroud truly dates to the Middle Ages, someone would have needed to understand the principles of photography hundreds of years before the invention of cameras. But photography wasn't developed until the 1800s. And the human eye cannot naturally perceive the world in negative form, let alone recreate it with such subtle shading and precision. This became the first major mystery surrounding the shroud. The cloth didn't behave like a painting. Instead, it seemed to behave like a photographic plate, capturing a single moment in time. Over the years, scientists studied the fabric using X-rays, ultraviolet light, infrared imaging, and laser scans. But surprisingly, the biggest clues were not found in the image itself. They were hidden in something much smaller. Microscopic dust particles trapped between the linen fibers during centuries of handling and travel. In 2015, a team of geneticists and biologists led by Professor Gianni Barcaccia from the University of Padua, received special permission to study these tiny traces. Their goal was ambitious. They wanted to examine DNA trapped within the cloth. The scientists were not trying to identify the DNA of a single individual. Instead, they hoped to reconstruct the history of the cloth itself, where it had traveled and who had come into contact with it across the centuries. To collect samples, the team used specially designed sterile micro-vacuum devices equipped with extremely fine filters. These tools gently collected microscopic dust, pollen, and organic fragments from the surface of the linen and from deep within the spaces between the threads, where ancient particles can remain trapped for thousands of years. They also studied small fragments gathered during a restoration performed in 1988, which had been preserved carefully in controlled conditions. The research was delicate. Even the smallest modern contamination, a breath, a sneeze, or a flake of skin from a technician, could overwrite the ancient DNA signals. The samples were taken into ultra-clean laboratory environments. There, scientists began analyzing them using next-generation sequencing, NGS, a powerful method capable of decoding huge amounts of genetic data. The researchers focused mainly on mitochondrial DNA from plants and humans. Unlike nuclear DNA, mitochondrial DNA exists in many copies inside each cell and tends to survive much longer in ancient material. It also traces maternal ancestry and can help reveal geographic origins. For weeks, computers worked non-stop, analyzing millions of genetic sequences and comparing them with global DNA databases. When the final genetic maps appeared on the screens, the researchers realized they were looking at something unexpected. The data did not show the genetic signature of a single individual. Instead, it resembled a genetic snapshot of humanity itself. The results were later published in the respected journal Nature Scientific Reports, and the reaction in the scientific community was immediate. Researchers had expected a dominant genetic signature. If the shroud had been created in medieval France, as some skeptics claim, then most of the DNA should have been European. If it originated in Jerusalem and remained there, then Middle Eastern DNA should have dominated the results. But that is not what the data showed. Instead, the cloth carried traces of people from across large regions of the world. Scientists identified genetic markers linked to several geographic areas. First, the Middle East. Certain haplogroups match those found among the Druze, a historically isolated community living in regions of Israel, Lebanon, Syria, and Jordan. Their genetic lines are considered extremely ancient and stable, making them strong indicators of Middle Eastern ancestry. Second, Western Europe. Haplogroups such as U5b and H1 to H3 appeared, which made sense because the shroud has been preserved in Europe for centuries, first in Chambéry and later in Turin. During that time, it was handled by clergy, restorers, and countless pilgrims. Third, North and East Africa. Haplogroup L3 appeared in the samples, suggesting connections with regions such as Egypt and Ethiopia, areas that hosted some of the earliest Christian communities. Fourth, South Asia. Haplogroups typical of the Indian subcontinent, including M39, M56, and R8, were also present. And finally, the scientists detected markers associated with East Asia, including haplogroups linked to China, India, Africa, Europe, and the Middle East, all represented within a single piece of linen. If the shroud were merely a medieval forgery created in a European monastery around the 14th century, it would be difficult to explain such a wide genetic mixture. The discovery suggested something else entirely. The cloth had likely traveled extensively across different regions before arriving in Europe. In other words, the shroud may not simply be a relic, it may be a traveler through history. When researchers mapped the genetic patterns geographically, they noticed something remarkable. The distribution closely matched an ancient route described in early historical writings, the path taken by a mysterious relic known as the Mandylion. According to Byzantine and Middle Eastern sources, the cloth was once folded in a way that only the face was visible, displayed inside a frame. This folded relic, sometimes called the tetradiplon, moved across several cities in the ancient world. The journey likely began in Jerusalem, the place associated with the death and burial of Jesus. From there, historical records suggest it was transported to Edessa during the early centuries of Christianity, and that is where the next chapter of the Shroud's mysterious journey begins. For centuries, the relic remained hidden inside the walls of the city of Edessa before it was eventually rediscovered. Edessa sat at a vital intersection of the ancient world, right along the Great Silk Road. Caravans traveling from China, India, Persia, and Arabia regularly passed through the city. Merchants carrying silk and spices, travelers, pilgrims, and diplomats all encountered the relic that many believed protected the city itself. People came close to it. They prayed before it. Some kissed the reliquary that held it, while others simply breathed near it. Each encounter left microscopic traces behind: skin cells, strands of hair, particles of sweat. Over centuries, layer after layer of these invisible biological fragments settled into the fibers of the cloth. Gradually, the DNA of countless individuals from across the world accumulated on its surface like a fine layer of dust. Eventually, the relic was moved again. This time, it traveled to Constantinople. From the year 944 until the early 13th century, it was kept in the center of the Byzantine Empire. Byzantine rulers either purchased or seized the relic and brought it to the imperial capital, which at the time was one of the most diverse cities in the world. Constantinople attracted people from every corner of the known world: traders, soldiers, pilgrims, and scholars. Then came the dramatic events of 1204. During the Fourth Crusade, the city was violently sacked and looted. In the chaos that followed, the shroud disappeared from historical records. Not long afterward, it appeared again, this time in Athens around the year 1205. After the fall of Constantinople, French Crusader knights came into possession of the relic. It remained in Athens for some time before eventually traveling west. By around 1353, the cloth made its first clearly documented appearance in Western Europe. It was in the possession of a French knight named Geoffroy de Charny. This historical journey creates a serious challenge for the theory that the shroud was simply a medieval forgery. A fabric created in 14th century Europe could not easily contain traces of DNA from places like China, India, Africa, and the Middle East, regions that most Europeans at the time only knew through distant stories of travelers such as Marco Polo. Even more unlikely is the idea that a medieval forger somehow predicted that scientists, centuries later, would analyze microscopic genetic traces and deliberately planted them in advance. The DNA on the shroud does not appear to be random contamination. Instead, it resembles a biological record of the countless people who came into contact with it over many centuries. It is like a silent archive of human presence, traces left behind by pilgrims, traders, and travelers who once stood before it. In this way, the genetic evidence suggests both an ancient origin and a long journey across the Eastern Mediterranean and beyond. But human DNA is only part of the story. Another important clue came from plants. The field of palynology, the scientific study of pollen, offered further insight into the cloth's history. Two specialists investigated this question independently. One was Professor Avinoam Danin, a well-known Israeli botanist from the Hebrew University of Jerusalem. The other was Swiss forensic scientist Max Frey, a pioneer in the use of pollen analysis in criminal investigations. Both scientists examined microscopic pollen grains trapped within the fibers of the linen. Using adhesive tape, they carefully lifted tiny samples from the cloth for study under powerful microscopes. What they found attracted enormous attention. In total, pollen from 58 different plant species was identified on the fabric. 17 of these plants grow naturally in Europe, which makes sense, given that the shroud has been preserved on the continent for several hundred years. But many others came from much farther away. Several species were native to the Middle East, Turkey, and the Anatolian region, areas that align closely with the historical route from Edessa to Constantinople. Then researchers discovered something even more remarkable. Some pollen grains came from plants that grow only within a very narrow geographic area between Jerusalem and Jericho. One plant stood out in particular, Gundelia tournefortii, a thorny desert shrub similar to a thistle. Pollen from this plant made up nearly half of all the samples collected from the cloth, a striking and unusual concentration. Why would pollen from a thorn-covered desert plant be so dominant on the fabric? Gundelia tournefortii grows in the region around Jerusalem and produces long, rigid thorns capable of forming such a crown. The plant also blooms during early spring, exactly the season when the Jewish festival of Passover occurs. The unusually high concentration of pollen from this plant, especially around the head and shoulder areas of the cloth, suggests the possibility that such thorns were present when the body was wrapped. Another plant provided additional evidence. Pollen from Zygophyllum dumosum was also found on the cloth. This species grows only in the Judean desert and parts of the Sinai Peninsula. Its presence reinforces the idea that the fabric had once been in that specific region. Normally, blood darkens to brown or black as it ages, yet the stains on the shroud appear unusually red. Scientific analysis found extremely high levels of bilirubin, a substance released by the liver during intense stress and physical trauma. This chemical reaction can preserve a brighter red coloration, offering yet another clue about the extreme suffering experienced by the person whose blood stained the cloth.